Dear students, now I am going to tell you about monoclonal antibodies. You have studied already the structure of antibodies and classification of antibodies. Antibodies are immunoglobulins are produced by plasma cells in response to an antigen or immunogen. Antibodies can able to bring specific immune responses. Specific immune responses means either they can able to produce antibodies or humoral immune response, or else they can stimulate the T cells and bring cell mediated response. So, antibodies can recognize and bind the specific antigen. So, antibodies bind to an epitope of an antigen. It can able to bind to a small portion of antigen, that is called antigenic epitope or antigenic determinant. You have studied already. So, this you will able to see in this this antigen, this is the antigenic epitope. So, that this antibody binds to this or recognize this and binds this epitope. This will bind this epitope, and you will see the antibody binding this epitope. So, this antibodies, this is IgG or immunoglobulin G. So, this can able to bind two antigens. You will find there are two antigen binding site for IgG. You have studied the structure already. So, this this is an antibody of bivalent that simultaneously it can bind to the two antigenic determinants. An antigen has several or many different epitopes and reacts with many different antibodies. So, this is what you can see, there are many different epitopes and many different antibodies are binding this. Okay. So, the same antigen. So, many antigens present numerous epitopes that are recognized by large number of lymphocytes. So, in single antigen having many epitopes, and there are many antigens, many epitopes circulating or getting to the body. So, they are recognized by the lymphocytes, and these lymphocytes they activate B lymphocytes, and the B lymphocytes are activated, and they will become plasma cells, and they start producing antibodies in response, is called polyclonal antibodies. Because so the single antigen having many antigenic determinants. So, these are recognized by different B lymphocytes, and they are producing different kinds of antibodies. So, the one B cell producing very specific to this determinant, another B cells producing specific to this for another for this. So, that is what you can see the in the body, the antibody responses or antibody production is polyclonal for different antigenic determinants. Polyclonal antibodies are antibodies that are derived from different cell lines, different B cells or different B cell B lymphocytes. So, they differ in amino acid sequences. Even though they belong to the same class, so for example, even though they belong to the immunoglobulin G, there are minor amino acid differences you will find with respect to the antigen binding site. Okay. So, what are monoclonal antibodies? So, monoclonal antibodies are antibodies, they are produced by single B lymphocyte. It is the clone of a single B lymphocyte, and it is producing only one type of antibody. It is very specific for a particular antigenic epitope. It is a monovalent. It can recognize only one type of the antigenic determinants or antigenic epitope. So, this monoclonal antibodies are produced for the first time, and by the technology called hybridoma technology by scientist, immunologist George Kohler and Cesar Milestone. Kohler and Milestone devised a technique called hybridoma technology for generating monoclonal antibodies for desired specificity for a particular antigen, very specific to a particular antigenic epitope. And for that, it is revolutionized the field of medicine. So it is a field of research, scientific research. There are these monoclonal antibodies are used in very different fields like medicine, for diagnosis, the therapeutics, industries, research. So that's revolutionized the field of science. So that's why they are awarded with the Nobel in 1984. So this is the hybridoma technology, that is they devised or they found to produce this monoclonal antibodies. What is this technique, hybridoma technology? We will study in later slides. Just you to understand what is the difference between monoclonal antibodies and polyclonal antibodies. Polyclonal antibodies are produced by different B cell clones or different B cell lymphocytes, whereas and they are specific, or they are for different antigenic epitopes, whereas monoclonal antibodies are produced by a single B cell clone. It is by a single B cell lymphocyte. It is the clone of lymphocyte, B cell lymphocytes, and they produce a specific monovalent antibody through the technology called hybridoma technology or hybridoma cell lines. What are these hybridoma cell lines? We will see. Antigen binding, how exactly? So heterogeneous antibody population, this is a different kinds of antibodies are produced here, one for this epitope, one for another epitope. So they are for multiple epitopes for a single antigen. Here it is the homogeneous antibodies, one kind of antibody. So it is you can see it is for a particular epitope you will find. Antibodies, you will find it is a mixed class of antibodies of different isotypes, whereas here a single class, it is a monovalent, very specific to bind with a particular antigenic determinant. The specificity is lower here because it is for different determinants. Here it is very specific to a particular antigenic determinant. Stability is high because if one antigenic determinants lost, also the antibodies will be there to bind with the other antigenic determinants. Here it is the low stability because it is if that particular antigenic determinant get lost, so that the antibody cannot able to bind. So what are monoclonal antibodies? Monoclonal antibodies are produced by the clone of B cells or B lymphocytes. They are very specific to a part of antigenic molecules or antigenic determinants and binds to that the epitope of that antigen. These antibodies or monoclonal antibodies are made by a clone or a particular immune cell, B cell clone. They are monovalent affinity. They can able to identify a particular antigenic epitope and it can able to bind on that. So these monoclonal antibodies are generated by identical B cells. So clone of B cells they derived from a single B cell clone. Single B cell, it multiplies because the B cell will multiplies and become plasma cell, multiplies, then they transform into plasma cells and they produce antibodies for a particular antigenic epitope. They can recognize the same epitope, all these antibodies. So these monoclonal antibodies are produced by hybridoma technology. So what exactly hybridoma technology? It is developed by Kohler and Milestone. So this monoclonal antibody production, it begins with the immunization of a mouse with an antigen. The first step, so the antigen is administered to the mouse. So this is called the immunization or administering the antigen to the mouse. after some time, after several weeks, antibody producing spleen cells or B cells are isolated. So we have to give some time. The antigen evoke the B cell proliferations in the spleen and they start producing antibodies. So for that purpose, after few weeks, then we have to take out the spleen and we have to isolate these B cells. So these B cells are isolated, but the problem here is the spleen cells, they do not survive in culture, they cannot multiply in culture. So that's why they have to be fused with some other, the cancer cells, they will multiply very fast in any medium. So that's why here this spleen cells fused with the myeloma cells. Myeloma cells is the bone marrow cancer cell. So the fusion of spleen cell with the myeloma cell, it gives rise to a hybridoma cell or hybrid cell. So you can see this is the myeloma cell. So this myeloma cell is fused with the spleen cell, and that is using a fusogen. Fusogen, what exactly, I will come to the later. So this is what you can see, the hybridoma cell is produced. This is by the fusion of spleen cell with the cancer cell, that is the myeloma cell, bone marrow cancer cell. So now this plate contains hybridoma cells. These hybridoma has the best properties of both the cells. So they can multiply very fast and also they can grow in the culture medium and they make antibodies. That is the advantage. They possess the properties of both the cells. So the cancer cells, they multiply fast, and the spleen cells, they are they can able to produce antibody for the injected antigen. Okay. So the fusing of B cell with the immunized animal with the immortal myeloma cell line done by a fusogen, that is called polyethylene glycol. Polyethylene glycol is it is a fusogen. It is used to fuse. It's like a gum like material, and it is used to gum the two cells. When the two cells come together, the two plasma membrane, so there will be the fusion reaction. That's why this agent, the fusogen polyethylene glycol, is used. So that's why in the cells under this condition, they get fused and they become hybridoma cell. So then we have to screen and we have to sort out unfused normal cells and unfused tumor cells. So all these we have to find out. B cells can grow in culture medium containing it is called the HAT medium. It is a culture medium, hypoxanthine amnopterin thymine in that HAT medium. But myeloma cells does not grow in HAT medium. So that's why unfused myeloma cells, they don't survive. Only fused myeloma cell will survive because it has got the properties of the B cell or the splenic B cell. So the myeloma cell fused with another myeloma cell also they cannot. So in this way we can able to screen. Only we can able to isolate hybridoma cells, those who are fused. So these hybridoma cell can survive and divide in the HAT medium. Then screening is to select hybridoma cells. They are producing our desired monoclonal antibodies. That also we have to find out whether they are producing the desired monoclonal antibodies for which we have administered the antigen. So these hybridoma cells are cloned and they are cultured to get large number of monoclonal antibodies. These hybridoma cells, they can be cultured at any time, and if we can freeze it and we can use it later, we can use after so many years, like that you can see we can maintain this cell culture for a long time, and huge quantity of monoclonal antibodies are produced continuously by this hybridoma technology. This is the overview. Any animal we can take, it may be the rabbit or monkey or mouse or rat, then we can inject with the antigen for which we want monoclonal antibodies. Then we have to isolate the spleen cells which are producing antibodies. Then we have to fuse with the myeloma cells, cancer cells. Now it is a hybridoma cells. Then we have to select these cells in the hat medium so that we can able to select only hybridoma cells. Then we have to select and screen with the hybridoma cells. They are producing the specific antibodies which is for which we wanted a particular antibodies for a particular antigen that can be screened. Then we culture only those hybridoma cells, and the excess cells we can be stored in the liquid nitrogen for long duration. So then we can produce these antibodies in any number. So this is the advantage of monoclonal antibody production technology, hybridoma technology. Now, what are the applications of these monoclonal antibodies? So, these monoclonal antibodies, the applications are, there are numerous. So, I cannot discuss all those. I just concentrate in this only the major one. So, the very first is it is the diagnosis. Now, all diagnostic tests for any pathogen, pregnancy test, all these they are using monoclonal antibodies. They are used, monoclonal antibodies are used in the detections of toxins, drugs, hormones, pregnancy test. So, the pregnancy test here, this is the in this you know the pregnant woman, even just she conceived, just after 14 days or 1 month, the test will become positive, because you know the pregnant woman urine contains, there is a the human chorionic gonadotropin. So this the test kit, it contains the monoclonal antibodies for this HCG, that is the human chorionic gonadotropin, that is present in the urine. So the urine is placed and then it will be, you can see what happens, you can see the urine contain HCG. So there is a immovable antibody, and when this antigen binds to this, and in the test site there will be a colored stripe will develop. So then it is a confirmatory. So if there is a two such things, then the test is confirmed. This is how the pregnancy test is now. It is with there is a test kit will come. Similar, you can see the diagnosis for cancer, allergies, and different diagnosis for leukemia can be done using monoclonal antibodies. So it is able to detect disease specific antigens. So the monoclonal antibodies are developed to find out this disease specific antigens, and it can be diagnosed. It is very quickly. That's why it's a rapid diagnosis. Within no time, one can able to find out the disease or pregnancy test, the disease diagnosis for hepatitis, influenza, herpes, streptococcal infection, HIV, all these. Now they are using these monoclonal antibodies to identify the strains of single pathogens, for example Neisseria gonorrhea, or else dengue, or else any which strain of a particular pathogen is infecting. That also we can able to detect with the help of monoclonal antibodies we develop specific for the antigen of that particular strain. So what are the advantage of the this as a diagnostic tool? It is a rapid testing, high sensitivity, and less material needed for this, and you can see the less expensive equipment. All this the advantage we have because of this monoclonal antibodies. So these monoclonal antibodies for the detection of pathogens, viruses, bacteria, protozoa, any pathogen or toxins can be detected by the technique developed is the enzyme linked immunosorbent assay, or ELISA. So they can be detected in a few minutes. you will find this is the basic the mechanism of ELISA. So these are immobilized antibodies present in the well, and when we add the sample for this, for example any sample what we are detecting, the sample, blood sample or urine sample or any sample, then you can see there will be, so the antigen will going to bind with the antibody, so that you can see it is further it will be linked with the another antibody which is which can develop a color complex, thereby we can able to detect whether a particular antigen is present or not, and we can able to detect the presence of the infection. So similarly, monoclonal antibodies in the imaging of the cancer, monoclonal antibodies and cancer detection, you can able to see the cancer tumor cells. So these you can see there is a monoclonal antibodies cancer detection based assay tests are developed now to detect the antigens and small molecules that are released by the cancer cells, and this make easier to identify cancer tumors in early conditions which can be treated and removed very quickly. So similarly, there will be radio immunodetection. So this imaging technique used to detect the presence of the cancerous or cancer specific cells that has been developed deploying radio labeled monoclonal antibodies. So these are all the applications. You can see monoclonal antibodies in therapeutics. So the monoclonal antibodies against the cancer, different types of monoclonal antibodies are used in cancer treatment. So this is it is as a therapeutic agent to treat cancer, to treat autoimmune diseases. So there are naked monoclonal antibodies in the market. So they are used to treat the cancers. They are in this commercial name, you can see. Then the conjugated monoclonal antibodies. Conjugated means now this is popular. Monoclonal antibodies joined with the chemotherapeutic agents or drugs or radioactive particles in the radiotherapy. So these monoclonal antibodies, they circulate throughout the body and hook into the target antigen, that is the antigen very specific expressed by the cancer cells, and delivers the chemo drug to that particular the cancer cells. These that thereby the damage to the normal cell will be minimized. Such chemotherapeutic drugs are developed now along with the use of these monoclonal antibodies. So such drugs are now you can see chemo drugs are rituxan, and these are all the commercial names. You will find chemotherapeutic drug, they are come along with the monoclonal antibodies. They can pinpoint where exactly the cancer cells. Similarly, radio labeled antibodies. Then there are bispecific monoclonal antibodies. So they are also developed. They can able to bind two different antigens and they are very helpful to treat acute lymphoid leukemia. Then monoclonal antibodies are used to treat autoimmune diseases. So the whatever the autoantibodies, so these monoclonal antibodies, they can bind and they can minimize the effect of these autoantibodies, and it will be helpful to treat rheumatoid arthritis, bronchial asthma, Crohn's disease, multiple sclerosis and other autoimmune diseases. So there are number of applications. So we can purify enzymes in research, hormones, nucleotides, proteins in the industries, and we can purify these molecules of enzymes, hormones, nucleotides, all these if it is present in a very low concentration in a medium. So that is the immunopurifications we can do with respect to interferon, membrane proteins, all this. Then we can able to fix the drug dosage or vaccine dosage, all these with the help of these monoclonal antibodies. We can develop vaccines they are helpful in raising specific vaccines for a particular strain of bacteria or viruses. Then organ transplantation, monoclonal antibodies are very useful in neutralizing the action of lymphocytes, which are responsible for the transplant rejections or graft rejections. So they also used it to destroy autoantibodies produced during autoimmune disorders. So they are also used for labeling a particular immune cells or cancer cells. They are used for the analysis of the complex antigen mixtures to isolate and purify. So there are number of other applications you can find out. So this is about the monoclonal antibodies and how they are produced through hybridoma technology and what are the applications.